Started. So welcome back to our virtual laboratory. During today's session, we are going to get to be biologists. Now, you may be asking, well, what does a biologist do? Biologists are people that study living things. And one of the main things that they look at is DNA. And so today's exercise, we're going to be actually getting to extract DNA from a strawberry. Now, you may have heard of DNA before, but what is that? Well, DNA stands for deoxyribonucleic acid. I know, a big word, but it's so important to us. When you think about deoxyribonucleic acid, or DNA, it's what makes us unique. It's why some people's hair is curly and others straight. It's why some people's eyes are brown and others are blue. It's what makes us unique. It's our hereditary information, and we get that information from our parents. We get a little bit from our mom, a little bit from our dads, and that's what makes each one of us unique. Now, DNA consists of over 3 billion pairs of these nitrogenous bases that make the code for who we are. Now, these nitrogenous bases, they are adenine, guanine, cytosine, and thymine. And they make different codes, different patterns to help determine who we are. So during today's activity, we're going to actually get to extract DNA from our strawberries, and we're going to have to do a little work to get that, because the DNA is found way deep in the cells, inside of the nucleus, that central part of the cell. A little bit is found in the mitochondria, but most of it is found inside of the nucleus. So in order for us to get our DNA out, we're going to have to first extract, then isolate, then observe it. Let's get started. All right, so if we want to extract DNA from strawberries, there's a few supplies that we need. First, of course, we need our strawberries. Now, we're using strawberries because they have a lot of DNA in them. They have what's called an octoploid, over eight different pairs that are there. We're also going to need some soap, some water, measuring cup and measuring spoon, a gallon-size Ziploc bag, some coffee filters. We're going to need a stick of some sort, whether that's a popsicle stick or a kebab stick like I have. We'll need a spoon, another spoon, some rubber bands, cups to actually capture our DNA, some salt, and some ice cold isopropanol. Now, you want to make sure you put this in the freezer the night before, or you can do like I've done and put it in some ice ahead of the time that you're ready to get started. Also, since we're working with some chemicals, some supplies, we're going to want to have our safety glasses as well as our gloves. Let's get started. All right, so we're ready to go ahead and extract our DNA. Now, remember, the DNA is found way deep inside of these actual cells that are in our strawberries. So the first thing we're going to need to do is try to get them loose. We got to extract them. So there's a few steps that we're going to have to do in order to get the DNA out of here. So first, you can go ahead and actually just remove that little green part from your strawberries. It's also important to note, if you don't have fresh strawberries, you can also use frozen strawberries. They work just the same. And you're just going to take your strawberry and place it inside of your gallon bag. Now, this is where you got to do a little work to actually break open those cells. So put it all the way down to the bottom. And then we're going to just start mashing it, all right? So the purpose of mashing is to give us access to where those cells are. Remember, the DNA is found way deep inside of those cells, inside of the nucleus, under a cell membrane. So we want to make sure that we can get access to them. So spend about a minute or so just squishing, trying to really break open those cells so we can get in there and get that DNA. So just mash it up. Move it all around. Just keep mashing, keep mashing. Once you feel like you've gotten all the big chunks of strawberry broken down, you'll know you're good to go. I'm just going to keep mashing. And you want to be gentle. Make sure that you don't puncture your bag, or else you'll get strawberry a little bit everywhere. I also forgot to mention that you're going to want to have a plastic table cover or something down just to capture any mess that may be created along today's experiment. All right, so that looks to be good. So you guys can see here that we have all of our strawberry broken down and we're ready for our next step. Now, as I said before, that DNA is found buried way deep inside of our cells. There's also proteins there. There is a cell membrane. There's all this stuff we got to get out of the way so that we can actually get to our DNA. So what we're going to make is something called an extraction mixture. Extraction means we're going to pull it out. We're going to take something out of where it's at. And so we need a couple of ingredients in order to make that extraction mixture. So you can take your strawberry bag and set it to the side for a little bit and grab an empty plastic cup. Now inside of that cup, this is where we're going to be making our extraction mixture. What we're going to want to first add is half a cup of water. So I have a little bit of water already here, and I'm going to get my measuring cup. So my measuring cup, this does work at half a cup. So I'm just going to slowly pour into my cup, my measuring cup, so I get about half a cup of water. All right, so once you have your half a cup measured out, go ahead and just add it to your cup. And once you've transferred all of that, you can take your water and set it back to the side. All right, so we got to add a few things to help us get our DNA out of those cells. One of the things that we're going to add to our extraction mixture is our soap. Now, why are we using soap? Soap can actually help to break down that cell membrane. Remember, the cell membrane is this oily lipid bilayer, and the soap has the ability to dissolve that layer away. And that's going to be critical so we can actually get our DNA out of there. So to our water, we're going to add 2 teaspoons of soap. So I'm going to grab my measuring spoons and I'm going to measure out 2 teaspoons of soap. Now this soap is able to dissolve that lipid bilayer, similar to how it works when we wash our hands. It's that property of like dissolves like. It has similar intermolecular forces to that lipid bilayer, so it's able to dissolve it away. All right, so that's 1 tablespoon, and we're going to add 1 more. All right. And once you got that in there, we're going to use our spoon just to mix that up. So to set this to the side one more time, grab my spoon and give it a nice little mix. Now you don't want to mix too much. You want to make sure that you are not creating a lot of bubbles. We just want to make sure this is a well-blended, homogeneous solution. Homogeneous meaning the same throughout. Okay? Now, once you've created your extraction mixture, we still need to add one more thing. Now, remember, inside of that cell, there is this cell membrane that we're going to be able to dissolve away with this soap. But there's also these proteins that are bound to our DNA. So we got to get that protein away from our DNA. What we're going to use is some salt or sodium chloride. The salt is able to actually punch holes into that protein and allow it to release the DNA that we want to get. Okay? So what we'll be adding to our extraction mixture is 1 teaspoon of salt. So I'm going to use my measuring spoons one more time and open my salt. This is just regular basic salt. We're going to pour that in so we have a nice leveled amount of salt in our teaspoon, and then we're going to add it into our extraction mixture. Okay, and we'll set that to the side. the beauty of having a nice clean tablecloth, so we can just clean that out of our way. All right. So once again, get your spoon and give it a nice stir. We want to make sure that that salt is well dissolved into our water and into our soap. Again, the purpose of this mixture is to be an extraction mixture. It's going to help us pull that DNA out of those cells. Okay, so just mix it up. And once you think all the salt is dissolved, should be good to go. Okay, I'm not seeing any salt particles anymore, so I think I'm nice and dissolved. Okay. All right. So we're ready to start extracting our DNA. Now what we're going to need to do is go back to your strawberry bag, and we're going to pour our extraction mixture right into our bag with the strawberry that we've pulverized. Okay. So open up your bag, and we're just going to gently pour into our bag. All right. Now this extraction process is going to need a little help. So we're going to have to actually seal this bag one more time. Try to make sure there's not a lot of air in there. So just squeezing it up. Make sure it's nice and sealed. And just move that around so that extraction mixture has a chance to work with those strawberry cells. Okay, I'm just going to gently squeeze with my hand, trying to make sure that that extraction mixture is doing the job. Remember that soap is dissolving the cell membrane, allowing us access to that DNA. And then that salt is punching holes into those proteins to free it up. Okay. I'm going to do this for about a minute or so, just working my way around the bag to make sure that we get as much of that DNA out of there as possible. Now we are using relatively safe materials, but it is always good to just make sure you have your personal protective equipment. That's why I have these gloves on, just to make sure we are not exposing ourselves to anything we don't want. Okay. All right. So once you've had a moment to allow that extraction mixture to work with that strawberry for a little bit of time, we're then ready to move on to our next step. So right now, you can't see it, but the DNA is actually floating around in this mixture. So what we're going to want to do is actually now isolate the DNA. We want to get it, we've extracted it from the cells, but now we want to isolate it. All right. So we're going to do a separation technique first to filter away some of the chunky parts of the strawberry that still exist to help us out. So set that to the side and grab another one of your cups. So I have another cup here, nice clean cup. Now to do the separation, what we're going to be using are coffee filters. Coffee filters work with helping us to separate mixtures that have solid material that's in them. So your coffee filter has small pores inside of it that allow smaller things to pass through and then captures those large pieces. And so this will be perfect for actually getting our extraction mixture away from our strawberry mixture. So what I like to use, I do like to use a rubber band to help me out here. And so we're just going to gently place your coffee filter inside of the cup, and then use a rubber band just to hold it into place. If you don't have a rubber band, don't worry, you can just hold your hand, but this makes it a little bit easier if you're working by yourself to do this process. So I'm going to try that one more time and get my rubber band onto my cup. Just put it right around that rim to keep the coffee filter in place. So again, that coffee filter has a lot of small holes in it. So the things that can pass through those small holes will, and the larger material will not. Oh, it smells so good, smelling the strawberries. All right, and so I'm gonna just gently pour this in to my cup. And you'll see it's gonna take a little bit of time, but immediately you can see that we start to see some liquid falling through our coffee filter. So as it's draining through, I'm gonna keep adding bit by bit all of this mixture so we can get as much of that DNA as possible into this cup. All right, and you can move it around just a little bit to try to help get it to go a little faster as we await our isolation process. Once a little bit is drained through, pour a little bit more in. So you can see this one gets a little messy, so definitely have your cup or your plastic table cover down. Again, I'm just moving it around just a bit. If you find that the coffee filter stops draining, that might mean it's a little bit full. There may be too much of the strawberry material that's in there, so you might have to swap in a new coffee filter, but we'll just let that process go. I'm just gonna keep adding a little bit more in, almost all of it. As you can see, it's just draining right in there. So again, this process is really just to help us separate our mixture. This is right now considered to be a heterogeneous mixture because you have the solid pieces that we can visually see as well as that liquid mixture. And so that is actually visible differences. And so we can use this separation technique in order to get them apart from one another. A little bit more. And try to get as much of that in there as possible. If you can't get all the foam out, that's okay. There's so much DNA inside of strawberries that we'll have plenty to view. All right, so get it all in there. Okay. Once you've gotten everything you can out of your bag, you can go ahead and set that to the side and use a spoon to help you to get a little bit of that liquid through a little faster. So all I'm gonna do is take my spoon and just gently press against it so that I can help to get a little bit more of that liquid through the cup, into the cup. So just very gently pushing against some of the strawberry pieces that are there to allow it to continue to drain. Okay, so it's going to take some time. Now, the other thing you can do as you are thinking about how this experiment works, you can think about other fruits that you might want to use. There are a lot of different fruits out there, but we need fruits that are mashable. Things like berries, cherries, blackberry, blueberry, any of those, bananas work well. You can try oranges, but you can see which fruit would actually give you the most amount of DNA. That's a really, really great extension to this project and really great science fair activity if you wanted to present something. Now, inside of your body, each human, there are over 3 billion pairs of those nitrogenous bases that make us who we are. And about 99% of those pairs, the way it's sequenced, are the same across everyone. But it's that 1% that is what makes us unique. All right, so it's still draining. Now what I like to do, once I start seeing it's taking a little bit more time, you can actually remove your rubber band and gently squeeze to help get this liquid through. And so I'm going to hold my cup here and very carefully lift up my rubber band. And that way I can grab the sides, make sure it doesn't spill over, and grab the sides of my coffee filter And this will allow me to just kind of squeeze it to help get that liquid through. Okay, so again, making sure I gather all the sides. And now this is going through a lot faster. Okay, so just keep that there. Give it a gentle squeeze, not to pierce the coffee filter, but this will help get as much of that out of there as possible. This is also why it's good to wear gloves, so you don't get any of this on your hands. All right. So we're just about got all of it out, and we'll have plenty of DNA to see. All right. I think we're good. Once you've gotten all of that of the mixture through, you can set that coffee filter to the side, and we're ready to actually see our DNA. Now, believe it or not, our DNA is in there. But right now, we still haven't completely isolated it. Right now, the DNA is floating around in that extraction mixture with that soap, the salt, the water. So we need something to actually get it out of that solution. We want to do something that's called precipitate. Precipitating it, making it precipitate. Think about what happens in the sky. When things precipitate, it means it's crashing down. It's falling out of the sky. So to help us precipitate this DNA, we're going to be using some of our ice cold isopropanol or rubbing alcohol. Now it needs to be ice cold so that we can keep that DNA intact. If it's too warm, it's not going to allow it to crash out. Okay? So what we're going to need to do is we're going to measure out into our cup half a cup of our isopropanol or our rubbing alcohol. Again, it was in the freezer overnight. And now I have cat it on ice, so it's still really, really cold. And so I'm gonna pour out half a cup of this, and we're gonna add that to our strawberry mixture. So measure out half a cup. And it's really, really cold. Okay. And we're just going to add this slowly into our strawberry mixture. So I'm just going to slowly add it in. Let that just kind of trickle to the side. Now, because the isopropanol has a different density than the water that's here, a layer is going to be formed. Just like how oil and water, you get that layer that forms. And our isopropanol is now able to sit on top of our water mixture. Now, if you can see, there's all this white stuff that's starting to form. That white stuff is our DNA. It's this long, fibrous material. Probably not what you would ever expect that DNA looks like. Now, after you wait a few minutes, you'll see this nice white layer starting to form. We need to get that DNA out of there. So this is where your stick will come in handy, okay? Now what you'll want to do with your stick, whether it's a popsicle stick or if it is a kebab stick like I have, we want to just very gently drag this stick in just the top layer. Make sure you're not going too deep into that bottom layer. Just drag it around in that top layer, all right? And so when I start doing that, you can see it starts to move. Now I'm gonna just start, because DNA is very sticky, I'm just gonna start rotating my stick around, and that's gonna allow me to start gathering all that DNA. All right, and so I'm just wrapping it around, to try to pick up as much of that as possible. But in just a few minutes, we've been able to grab our DNA fibers. Kind of looks kind of weird. It's definitely not what I would expect DNA to look like. But a great exercise that you could do is to try doing different fruits to see how much DNA are you able to capture from your different fruits. And remember, all of this was found in just one single strawberry. All right? So, I hope you all enjoyed today's activity. Remember, we learned all about biology and DNA, or our deoxyribonucleic acid. We learned that DNA is what makes us unique. It's our hereditary information. It's where we get everything that makes us us. We learned about what we need to do in order to get the DNA out. We have to first extract the DNA, and we did so using some salt to punch those proteins down, and the soap to actually break down our cell membranes. We then had to isolate by using a separation technique, the filtering, and then precipitating out our DNA. So I hope you all enjoyed today's activity. Can't wait to come back and see you all again in our virtual laboratory.